Namaste. I am JVN. I am Dr. Anupama Goyal. I am working as Associate Professor in Botany in Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to deliver my video lecture on Saccharomyces, which is a fungus, and this lecture on Saccharomyces will be useful for the students doing graduation as well as post graduation. So I start with Saccharomyces. Well, first of all, I explain you about the systematic position and classification of Saccharomyces. So Saccharomyces belongs to the kingdom Mycota, and the division is Eumycota, and the class is Ascomycotina. So Ascomycotina is the class in which the Saccharomyces has been placed, and the subclass is Hemiascomycotina, and the order is Endomycetes, and the family is Saccharomycetaceae. Family is Saccharomycetaceae. So the genus we are talking about, Saccharomyces. Saccharomyces is represented by many saprophytic species which are found ubiquitously. Now the question arises, what is Saccharomyces in Greek? There are two words. One word is saccharon, and one word, as you see, is the saccharon. This is the saccharon. It means sugar. And the another word is myx. This means fungus. So Saccharomyces are mostly found in media which is rich in sugar, or the Saccharomyces fungus is found in organic matter of vegetable origin. So there are many species of Saccharomyces. One of the most important species of Saccharomyces is Saccharomyces cerevisiae, and the Saccharomyces cerevisiae is commonly known as river yeast or baker's yeast. So what is the common name of Saccharomyces cerevisiae is baker's yeast, and this baker's yeast is used widely in wine and baking industries. Now this Saccharomyces produces two types of enzymes. So the name of the enzymes are invertase. Here is the invertase and zymase. So it produces two types of enzymes, an extracellular invertase and an intracellular enzyme called as zymase. So the invertase hydrolyzes cane sugar into dextrose or invert sugar, and zymase enzyme breaks down the invert sugar into ethyl alcohol and carbon dioxide. So this is the zymase enzyme. It breaks down the sugar into ethyl alcohol and carbon dioxide, and now they easily destroy soft cheese and similar foods, and food items are destroyed by Saccharomyces, and for this they impart repulsive yeasty flavor. Now next, what is the vegetative structure of the fungus Saccharomyces? So as you well know, the vegetative plant body is thalloid in nature. It is thalloid plant body, and this plant body is unicellular. It is made up of only one cell, but sometimes due to the formation of pseudomycelium or false mycelium during rapid multiplication by budding, and cells remain attached in chain. So because of the formation of pseudomycelium, the cells are remaining attached in chain. The cells shape, they may be globose, the cells may be elliptical, or the cells may be oval to even rectangular. So the cells may be globose in shape, elliptical, and the size of the cell ranges from about 5 to 6 times multiplied by 6 to 8 micron meter. The Saccharomyces cerevisiae shows that the cells are surrounded by a distant cell wall with three layers. The outermost layer consists of protein and mannan. So the outermost layer is made up of protein and mannan and some chitin, and the middle layer is made up of glucan. It consists mainly of glucan. The middle layer consists of glucan and the innermost layer consists of peptidoglycan. Now, this is the vegetative structure. So the cellulose is absent in the cell wall. As you know, in algae, when we study algae, in the algal cell wall, cellulose is present. But in Saccharomyces, cellulose is absent in the cell wall. Inner layer next to the cell wall is plasma membrane, also called as plasma lemma, which has series of shallow elongated peaks or invaginations. So plasma lemma is showing various invaginations in a large vacuole. It limited by a single membrane called as a tonoplast is occupying the center of the cell. So the vacuole is found in the center of the cell in Saccharomyces, and it contains a watery substance and granules of polymetaphosphates and lipid, and a minute double unit membrane nucleus is present near one side of the vacuoles. So this diagram shows the cell structure of Saccharomyces cerevisiae. Here in this diagram you can see the cell wall is there, and this cell wall is not made up of cellulose, and it is made up of three layers, and it contains a nucleus. Here it is well seen in the diagram. The nucleus has a nucleolus, and the the nuclear membrane, as you see in the diagram, is perforated by nuclear pores. So this shows the nuclear pores. There is a well organized nucleus, as you see in the diagram, and there are various mitochondria, as you see clearly in the diagram, and a large vacuole, as I have previously told, is very clear here in the diagram. This is the vacuole, and this vacuole, as you see in the diagram, is surrounded by the membrane, and the membrane is called as tonoplast, and some storage granules are also found to be scattered in the cytoplasm of the Saccharomyces cell, as you see in the diagram. Now, how the vegetative reproduction occurs in Saccharomyces. Now, vegetative reproduction takes place by means of fusion and budding. So, what is fission? Fission also, fusion or fission. First of all, fission. In a favorable condition, a constriction and transverse wall develops in the cell, and the cell splits into two daughter cells. So fission takes place when the conditions are favorable and a cell splits into two daughter cells, and this process is called as fission. During fission, a constriction appears in the middle of the cell. There is the appearance of the constriction in the middle of the cell, and simultaneously nucleus also undergoes mitotic division. The nucleus undergoes mitotic divisions, as a result of which two equal sized uninucleated daughter cells are formed. Here is a diagram showing how fission takes place in Saccharomyces cerevisiae. Here you are seeing there is one cell, and means Saccharomyces is unicellular. Then a constriction starts appearing in the middle. So you see, this is the constriction which is appearing in the middle, and then this constriction enlarges. So you see, from a single cell, two daughter cells are formed, and I told you the nucleus undergoes mitotic division, and you see the two nuclei also there. And now these two cells later on separate with each other and they form a new thallus. So now budding, what is budding? It also takes place during favorable conditions, means when the conditions are favorable, budding occurs in the fungus Saccharomyces. In this process, a small outgrowth protrudes out from the vegetative cell of the yeast, and the protoplast of the cell is transferred to this small swollen part. After mitotic division, two nuclei are formed. Here mitotic division occurs and two nuclei are formed. One of the nucleus is transferred to the outgrowth and the other nucleus remains in the mother cell. The size of the bud is always smaller than the mother cell. After maturation, these buds separate from the mother cell and leave a convex scar on the surface, which is called as a bud scar. Similar scar with concave surface remains on the wall of the bud, called the birth scar. Now you see, this is the diagram. The Saccharomyces cell is undergoing the process of budding. So you can see here, these are the buds. The Saccharomyces is reproducing by the formation of the buds, and here you can see the bud scar is also here, and this bud is formed at the top. So this is the mother cell and this is the daughter cell. Now budding. Sometimes due to the rapid division, large number of buds develop without being detached from one another and persist in the form of branched or unbranched chain and give the appearance of false mycelium called as pseudomycelium. Means when a large number of bud develop, they do not get separated from the mother cell or from one another, as a result of which unbranched or branched chain is formed and it gives the appearance of a false mycelium called as pseudomycelium. Finally, the cells get detached and grow individually. Now, asexual reproduction in Saccharomyces. How asexual reproduction occurs? It takes place by means of endospore. Well, in this process, the thick walled spores are formed, and these thick walled spores are called as endospores. So endospores are thick walled spores. It happens in the unfavorable conditions. Well, the formation of the endospores takes place when the conditions are unfavorable, and during this process 4 nuclei are formed. There is a formation of 4 nuclei by the mitotic division. The protoplast divides into 4 units, each with 1 nucleus, and so it forms 4 endospores. So 4 endospores are formed on coming of the favorable conditions, or when the favorable conditions arrive. Endospores germinates by budding and buds grow individually. Now I take the main point, how Saccharomyces reproduces sexually. So sexual reproduction in Saccharomyces cerevisiae. So in Saccharomyces cerevisiae, sexual reproduction takes place when the conditions are unfavorable. In this genus, it is very simple process because of entirely absent of sex organs. Means in Saccharomyces, which is fungus, sex organs are entirely absent. Then you will see how the sexual reproduction is occurring when the sex organs are absent in Saccharomyces. Then two somatic cells here I underlined it. Two somatic cells or two ascospores are involved, which assume the function of copulating gametangia. Two such cells come close up and develop beak-like outgrowth towards each other. Both the outgrowths come in contact and dissolve the wall of intervening. So the nuclei of both the gametangia, come to the fused outgrowth, that is in the conjugation tube, and diploid zygote produced after their fusion. Okay, and now in the next class, thank you, I will explain you in detail how the diplobinding haplobinding cycle occurs in Saccharomyces. Thank you.